it extends throughout the Gulf of Mexico coastline there. So just to orient you a little bit on tree rings, since I'm not necessarily talking to a dendro community, we could cut down a tree and see the rings inside that tree. And each one of those rings represents a year of growth. And as those rings change in size, it represents the environment in which that tree grew. So a very small ring might indicate a drought year, or a very big ring might indicate that the tree grew for a long time during that year. But instead of having to cut down the tree and get what we call a cross section, instead of having to do all of that work and kill a tree, we can just take small increment borers. So you can see in this image here to the right, a colleague of mine, Tommy Patterson, is holding one of these cores in his hand. And eventually we process those cores to the image at the bottom of your screen that you see here. So a tree core that's been mounted and sanded, you can see all of the rings of this pine tree in this core. You can also see each light and dark band combination is 1 year of growth. So you can see this tree goes back to 1945, year 1945. And there are periods of growth where there's much less growth here in the middle of the core, and that changes throughout its lifetime. We can measure the size of these rings and get what we call a chronology. Multiple trees, if we take just if we use just one tree, essentially, we're going to get a very biased picture of how the environment interacted in that area, because the tree might have been affected by disease. It might be genetically altered itself through evolution to more easily withstand different environmental stressors. So we take multiple cores from multiple trees, and then we average them out. So this line graph here at the bottom represents the stand of trees that you saw in the image earlier. And each one of the colored lines represents the width of each ring from the trees. So as we start in 1910, there's only one tree there, and we'll talk about this in a moment, because only one tree was growing in 1910 in this stand of trees. These had been logged, so it's not unusual to see that they don't go much further back than that. But as we get further down the line and further towards more recent years, we start to add some trees in this group. And eventually we can get an average, and you can see that these lines dip in different places as well. So we can plot these averages, and eventually we can note where hurricanes occurred. And for this particular stand of trees, after every single hurricane on the record in this area, we had a drop in growth. So and some of the biggest drops were some of the biggest hurricanes on record. So 2005 was Hurricane Katrina, a pretty big hurricane in the area. The advantage to using tree rings is that we can go very far back in time, much of the time, and we can say perhaps a hurricane occurred here. It's not on the record, but perhaps a hurricane caused this huge drop in growth for these trees at this time. So just to summarize, the quick talk here, 5 minutes talk. I love lightning talks. Basically, coastal tree ring research works. So we can use coastal tree rings from a geographical perspective, from a climatological perspective, and give long-term ecological gains to the ecology of the area and to the ecological community of the area. And the other thing that we can glean is what is actually causing stress to plants here. And so, for example, these trees here are stressed by coastal storm surge. Salt water hits these trees and it stresses them out. They don't have fresh water that they need to grow. And so salt water is certain, from storm surge specifically, is certainly a stressor to large plant growth. And it's probably also a stressor to smaller plants in the area, telling a story for the whole ecosystem here. If you have any questions about dendrochronology or tree ring research, or I'm going to do a sad plug here, if you have big trees anywhere that you study in wetlands, I'd be glad to sample them at some point. One of the exciting things about this virtual conference is that it's international, and I'd love to do some international coastal dendrochronology someday. So I work mostly in the coast, but if you have any questions or any comments about where you work, please feel free to email me. Thank you very much.
All right, so the title of my talk is Tidal Marsh Response to Sediment Deposition from Storms, and this is part of my thesis research at Villanova University for my master's degree. So just a brief introduction to what I'm talking about a little bit. So my study site was in Massachusetts at the Plum Island Ecosystem LTER. If you're aware of that program, they're long-term ecological research stations, and so a lot of research has been done there. And I was looking at several different kinds of storms, and those different storms can create different types of sediment deposition. So just a brief look at some interactions between these storms and the salt marshes. So one of the ecosystem services that salt marshes provide is storm surge mitigation. And that is due primarily to the vegetation on the salt marsh. And you can see in those pictures, the Spartina alterniflora, which grows primarily in the low marsh, so at lower elevations, and Spartina patens, which grows in the high marsh. There are also damages that storms can cause. I think this is more public knowledge, I guess, in terms of erosion, as well as excessive flooding. And then what I was looking at were more possible benefits. So the sediment deposition from these storms can help the marshes accrete vertically to keep up with sea level rise, and may also bring nutrients to help plant growth. So my research question was, what is the impact of the storm-derived sediment deposited on tidal marshes? And for that, I set up plots under different conditions. And so these are my conditions. So I looked at 3 different seasons, so autumn, spring, and summer. And then for each of these seasons, I looked at the low marsh versus the high marsh with their plant communities that I mentioned. And then I looked at 2 different types of sediment, a muddier, finer sediment and a sandier sediment, and 3 different depths of deposition, 2 centimeters, 6 centimeters, and 10 centimeters. So for this particular talk, I'm looking at 2 of the things that I collected data on. One was just elevation change. And so I used elevation pins, which you can see in this picture, sticking up through the depositions, to measure the change in elevation after the deposition was put down. And also I looked at above ground biomass. So what biomass was able to grow through these depositions after they were put down. So here is an example of one of my results, one of my graphs of results for the elevation data. So if you look on the y-axis, you can see the 10 centimeter, 6 centimeter, and 2 centimeter depositions, as well as a control. And you can see here that the control gained elevation, that there was natural deposition over the course of this. This was the autumn deposition, so over the course of a year. And you can also see that particularly initially, there was erosion from, and the more erosion occurred, the higher the initial deposition. And that was a trend that occurred pretty much everywhere. You can see here in the low marsh that initial erosion was relatively significant. Whereas if you look at this graph of the high marsh here, you can see that there is still erosion, but it is not as much, that the low marsh lost much more. And that is presumably due to the fact that it floods every single high tide, whereas the high marsh does not. So the other thing I wanted to talk about in terms of this elevation was looking at the different seasons that I mentioned. So this graph was made using a rate of change in elevation. So again, you can see here with the control, this is the control, that it gained elevation, and then with the different treatments over here. And the trend I want you to notice here is that the autumn deposition in yellow generally had the most loss, whereas the summer deposition in gray generally had the least loss. And that is presumably due to 2 things. The autumn deposition had winter weather immediately after the deposition, and had the least biomass. In the summer, you have mostly nicer weather and significantly more biomass. So the one other thing I wanted to talk about was this biomass. And so this is the Spartina patens in the high elevation. And you can see in this graph that the larger depositions really did have a very significant effect on the amount of biomass that was able to grow through. And you can see in this picture, this very heavy mat of Spartina patens, but it wasn't able to come through the deposition in here. And that trend was seen in the high elevation marsh with patens, but was not seen in Spartina alterniflora in the low marsh. So that just brings me very briefly to my conclusions, which are that the depth of deposition affects the amount of erosion, that the season of deposition also affects the amount of erosion, and that the location within the marsh makes a difference, both to the erosion, where there's more erosion in the low marsh, and to the biomass, because Spartina alterniflora is a much more resilient plant than Spartina patens, which had a greater mortality. So very briefly, since I ran over a little, I'd like to just thank Villanova, all the people who helped me, and the funding I received. Thank you.
Hey everyone, I'm Wendy Ampuero Reyes. I'm from Peru. So I'm going to talk about a little bit of my research when I was like back home doing my undergraduate studies. Right now I am a master's student at McGill University, so I am enjoying right now the summer of Montreal. So I'm just going to share my screen. Just give me 1 second, please. Okay, so can you see?
Yep, you are good to go.
Okay, awesome. So I'm going to talk about the carbon storage of Conoplectus americanus in coastal wetland Pantanos de Villa, wildlife refuge in Peru. So this is a picture of the field site. So it's important to mention that this is a salt marsh. It's a protected natural area, and also it's a Ramsar site. Okay, so in the photos you can see this plant species is a Conoplectus americanus. So it's known, well, in Spanish, junco or juncal. And on November 2014, there was a fire disturbance, lasted 12 hours. I remember that because I was on my way to the university, and my university is nearby, so I was able to see the fire, and it affected almost like 7 hectares, and of course damaging flora and fauna of this site. So we wanted to compare the carbon stocks in the burned and unburned areas of these dominant species after 1 year of the fire. Also, it's important to mention that the Conoplectus americanus is a typical species of the region, usually along the coast of Peru. And so regarding to the field site, so Peru is located in South America, and this field site is located in Lima, which is the capital city. And Lima is the province, so we have also metropolitan Lima. And here we have like almost 50 districts. So one of them is Chorrillos, which this Pantanos Villa is located. So as you can see, this is a small map of the area. So the green area is regarding to only Conoplectus americanus species, the junco. Okay, so the fire, I don't know if you can see my mouse, but the pointer, but the fire was located in that area. So I was like trying to compare. I really wanted to compare the carbon stocks in these burned and burned areas. So after 1 year of disturbances. So regarding to the methodology, it's like we hardly located like 5 plots of 1 meter square, as you can see here, and collected like 3 soil cores in each plot. So for a total of 15 cores per treatment. These soil cores was collected like to 30 centimeters depth and sectioned into 10 centimeters intervals in the field. So it will be like from 0 to 10, 10 to 20, and 20 to 30 centimeters. And also we did this core sample with negligible compaction. So in the lab, I separated the roots and rhizomes from the soil matrix, and usually, and then we calculated carbon storage using an elementary analyzer. So it's also important to mention that we wanted to follow the Blue Carbon Initiative methodology. I don't know if you're familiar with that, but it's like we wanted to estimate the carbon storage in 4 carbon pools, also above ground and below ground. So as you can see in this picture, this is the living biomass. This is the leader biomass that you know what, it's like we found a lot, a very huge amount and burning area. And this is the roots, rhizomes, and this is the soil matrix that we estimated carbon. So the unburned area is almost like 20% of the total area of Los Pantanos de Villa. And the burning area, I know that's a very small amount, like 1%, but because of the fire, it was almost like 7 hectares. So this burning area represents like almost 50% of this total disturbance. So our results. So this graph shows the average of carbon pools in unburning and burning March. So we have the average above from here, the below from here, and also we separate the roots and the soils with every 10 centimeters intervals. So from this graph, as you can see, all the unburned areas are higher. And as I mentioned, we didn't find any litter in the unburned area, even because we do this field work like after 1 year of the disturbance. And as you can see here, the carbon stocks in the burning and the unburning area decrease. As you can see, decrease with that. And also in the burned area, as you can see here in this graph, is like it's substantially lower compared to the unburning areas. And also the soil store, as you can see here, is going in the 10 centimeters. So the soil store the most significant amount in the first 10 centimeters compared with the other carbon pools. So at the end, we also wanted to estimate a CO2 stored by this species in Los Pantanos de Villa. So as you can see here, it's like the stock in carbon in the unburning area, or we can say that natural growth of Conoplectus americano is like 60 times greater than the burning area. And also we can say that Conoplectus americano March, also March, is a potential carbon sink compared favorably to global average, but also are substantially reduced when burned. So unfortunately, here in Peru, this kind of disturbance in coastal wetlands occur at least once per year, and most of these fires are of anthropogenic origin. So it's like, I think that we need to make sure, make more conscious and awareness about to protect these areas. So I think that I took my 5 minutes, and if you have any questions, please just let me know. This is my email, and you can just send me any questions. So if you want to know a little bit more about this field site, I'm glad to answer any questions that you got.
Well, thank you so much. My name is Anna Pushkov. I'm a master's student at the University of Connecticut, and I'm working with Dr. Beth Lawrence on coastal wetland restoration techniques, specifically the experimental thin layer placement method. So we're interested to see how do plant and soil responses respond from different sediment thicknesses. So just a quick background, we all love wetlands, but coastal wetlands are particularly valuable because they provide ecosystem services such as carbon storage and critical habitat. And the vegetation in these zones are controlled by the flooding regime, where the lower, more flooded areas are dominated by Spartina alterniflora, and the mid-marsh by Spartina patens. And this flooding regime is really important for maintaining surface elevation. As the high tide comes in, it deposits sediment onto the marsh that's slowed down by the presence of vegetation. And as that vegetation dies, it contributes organic matter that sediment can further deposit back onto, helping the coastal wetland keep up with relative sea level rise. But in the case of accelerated sea level rise, accelerated sea level rise can outpace this building feedback mechanism, where essentially coastal wetlands will drown or submerge. And the vegetation needs to be present there in the first place to allow sedimentation to occur. But this is also affected by the amount of sediment that's within a system. And as sea level rises, higher areas of the marsh will become more inundated and more favorable for Spartina alterniflora that will push Spartina patens further up to coastal development, reducing high marsh, mid-marsh area, otherwise known as the salt marsh squeeze. And the excessive inundation in the lower areas will reduce plant growth as phytotoxic sulfides build up in the soil and kill vegetation. So one way, so how can we help drowning coastal wetlands? So one method that has been used more extensively in the Gulf Coast, but in more recent years, the Atlantic and New England research coming up, is thin layer placement, which is a method that manually adds sediment to the marsh, immediately increasing elevation. It helps increase redox, which can help reduce the amount of phytotoxic sulfides that build up and promote plant growth. And this method, while, like I mentioned, has been extensively used in the Gulf Coast, it has not been tested in Connecticut. And after consulting local wetland managers, they're interested in how thick is too thick, because if you add too much sediment, you may smother plants, or if you add too little, it might not be cost effective for projects. So that brings us to our question: how did differential sediment addition affect plant and soil responses? So in May 2019, I set up a randomized complete block design where plot enclosures of 1 by 1 meters received either no addition, low level amount of addition, medium amount of addition, and high. These were put in plot enclosures that allow the sediment to stay in place, but water to move freely throughout, so it had the most natural hydro period. And then a control to the control, where there was no plot enclosure and no sediment added, to see if the plot enclosure had any effect. The replicates were blocked by elevation because of heterogeneity throughout the marsh, and sediment depths and water level were monitored hourly. We collected a suite of vegetation and soil responses, specifically interested in shoot biomass, root density, stem density, and height, as well as redox features and decomposition in soils. So this is just a visual representation of some of the responses, but as you can see, the increasing sediment addition decreased percent vegetation cover. We actually had 0 plants grow in any of the high treatments, and in the medium treatments, we only had three of the 6 replicates have a vegetation response. In contrast, for stem height, we saw with an increase in sediment, it increased stem height. I didn't show it at the high treatment at all because no stems grew in those treatments, but for reference, the plot enclosure itself is 30 centimeters tall. So you can see how after just one growing season, plants were increasing in height, but decreasing in cover. So looking at both of them together, how does the effect of sediment addition affect stem density and height? Increasing a sediment addition appeared to increase in cover while decreasing in density. Specifically bringing your attention to the low treatment, where the low treatment was different letters denote statistical differences, and the low treatment had a lower stem density and a higher stem height. maybe this may have made up for the increase in height, may have made up for the decrease in cover, as far as it's interesting to think about the allocation of resources that the plants may have when put in these maybe more stressful conditions for the short term. Combining stem density and stem density and stem height, we also calculated total above ground biomass. And what's interesting here is that in the low treatment, there was no difference between the controls and the no addition. So there may have been a difference made up. This is just reiterating the point where the stem height and the low may have made up for the difference in stem density when it comes to physical actual biomass. Looking below ground, we took 2 soil cores, one in the native marsh soil and one in the sediment that was applied, which is why in the sediment applied graph there is only 3 box plots, because there was only, those were the only treatments that actually received sediment. And the low treatment, interestingly enough, has the highest root density both in the native soil below and not as surprising in the sediment applied because of the above ground biomass response, but it's really interesting to also point out that while no stems grew in the high treatment, there was some root density, some root growth, both in the medium and the high. So really excited to look at the responses this year and how, if they make it back through. I measured redox using IRIS, which is a method where iron oxide is painted onto PVC films, and then in reducing conditions, soil microbes use that available iron oxide as a terminal electron acceptor where it's removed. So you can see in the soil reducing conditions decrease with increasing sediment, which is what we had expected to create more oxidizing conditions to hopefully promote root growth and less stressful conditions for plants. Looking at decomposition, I used a method called the TBAG index. It uses a standardized substrate for measuring decomposition, so you can compare decomposition rates across ecosystems. And looking at, we didn't see any differences across most treatments except in the high treatment, which when you pair that with the redox data, it's really interesting to look at how the treatment affecting redox affected soil organic matter decomposition, where more oxidizing conditions promoted more soil respiration and decomposition. So some conclusions and next steps. The low treatment seemed to be optimal for vegetation and decomposition, as decomposition rates were lower. And really excited to check out this year's data. As I went back to look at my site just like a week or two ago, and there were some stems growing in the high treatment where we had none last year. So maybe more above ground vegetation will grow, and we'll have some more responses in the medium treatment as well. We'll also be looking at other carbon dynamics, including CO2 fluxes and sulfides, where looking at how do these treatments also affect carbon dynamics and the sulfidic stressors that may be explaining some of the growth. And ultimately, the thin layer placement of sediment may be a viable option for restoration in the Long Island Sound. We will be monitoring these plots long term. And I also really look forward to all of the other work coming out of New England on this topic. So with that, I will take any questions. I'm going to say thank you to all my funding sources, including the SWS. So thanks.
Okay. So hello, everyone. My name is Whitney Siskoyas. I'm an undergrad studying at North Dakota State University. I'm really happy to be doing this today, so thank you to everyone who's listening. Today I'll be discussing if it matters if soil pH is measured in situ or in the lab after drying. So typically measuring the pH in wetland soils follows past methods that adhere to agriculture, yet they don't necessarily work for wetland soils. For example, when you dig up wetland soil and you dry it, you oxidize it and the pH should change significantly. Croplands, however, are already oxidized due to being tilled. Therefore, when you dry these soils, the pH shouldn't change. So all of the previous measurements are developed for cropland soils, but they're still being used to study wetlands. So my question is, the pH in wetland soils is often determined in the lab after soils have dried, but is that representative of the actual pH in the field? So therefore my goal is to determine how drying wetland soils will affect the pH. And overall, just to define soil pH testing methods so that they adhere to wetland soils specifically and not just the past methods that were made for agriculture. My hypothesis would be is that drying wetland soils should lead to oxidation, which would generate protons and lower the pH. So as far as my methods go, I measured soil pH in 25 wetlands in the field, after which soil samples were collected and dried in the lab. The majority of samples were taken from the Prairie Pothole region. Samples were dried in an oven until constant weight. pH was then determined in a 1:2 soil paste. Interestingly, you can see that pH in dried soils was generally higher than in fresh soils. You can also see a wide range of changes in pH from testing in the lab to testing in the lab after drying. These changes vary from a difference of 0.09 to a difference of 1.42. So this increase in pH from field to lab could be explained by the typically alkaline sodic characteristics of the wetlands in this region. Although I'm continuing to look into why it's increasing, especially when I did hypothesize that the pH would decrease due to it being oxidized. There's also this huge variation in how much the pH changed from field to lab, which varied from the 0.09 to 1.42, which is a pretty huge change when thinking about pH. As you can see, most of the samples pH increases, but there are 3 samples where the pH decreased as well, which is also good to point out. Even though early data do not support the original hypothesis, what's important is that we do see this enormous range in variation. As far as any future work goes, this is an unfinished project, and there are a few more things I'd like to look into. I've collected and analyzed soil samples from the 25 different wetlands, but I do plan on expanding my sampling range in order to have a more diverse group of wetlands to study. I'm also in the process of studying organic matter, as that can have a significant impact on pH as well. So if there are any questions, please feel free to contact me on Slack or throw a question in the chat box. I would absolutely love to go over any questions, or if anyone has any advice to give me or share with me. And I just want to thank John for putting this together, and I really appreciate this opportunity. So thank you.
So I'm from, as you can probably pick from the accent, I'm from South Australia. It's just gone dawn here, so it's fairly early. I'm an estuarine ecologist. I'm taking a mid-career break to do a PhD on the Coorong. Now, the Coorong is the traditional country of the Ngarrindjeri people, and so we always put here that it always was and always will be Ngarrindjeri Ruwi. And this is just, you know, estuary, gorgeous pictures of our wetland. It's 90 miles long. It has 5 different water sources within it. It is the largest wave-dominated estuary in South Australia, at the end of the 3rd longest navigable river in the world. It's also obviously Ramsar listed. we've got some major problems, and the reason I've stopped to do this PhD is that it's the home of a fairly large fishery. It was originally worth, in today's dollars, some hundreds of thousands of dollars, hundreds of millions of dollars. It's now worth $35 million a year to the local economy. But it's in what we call habitat-induced decline. So what's happening is every year the fish yield gets less and less. So down the bottom here, you can see a diagram of one particular species, which is black bream, which is an estuarine-specific species that has slowly, since 1985, declined to a point it's now a tenth of what its previous yield was. And when we start looking at the estuary, this is very obvious why. So the image behind is a chlorophyll A indicator. It's meant to be all blue or yellow. And down the southern end of the estuary, if you can see my mouse, you can see that it is bright red. In fact, the Australian geoscience people tell me it is the highest level of chlorophyll A in any estuary in Australia. And during summer, 80 kilometres of the estuary looks this bright yellow colour, which happens to be counterintuitively a combination of blue-green algae and golden diatoms. Large amounts of it are monosyphidic black ooze, and you have paddocks and paddocks of dead creatures, tube worms in this case, that occur during heat waves. So it's really, really looking bad. But one of the biggest challenges is the estuary, because it is so large, has been declining for a very, very, very long time. And something is occurring that we call shifting baseline syndrome. Now, I'm not sure how familiar people are with shifting baseline syndrome, but it's where our knowledge of what a system was like shifts and we forget what happens before. So usually when we talk about this, we talk about it in terms of generations. So this is your classical diagram that shows that it started really good at the beginning of the diagram, and then over a generation of usually very poor records, it declines a little bit, and then another generation of people comes along and it shifts further and further and further as the generations go on. Unfortunately, with the Coorong, it's fairly remote, and so what's happening is there's groups of scientists that come through, and they turn over a lot quicker than generations of the local community do. So the local community have one understanding of what the Coorong used to be like, and the scientists that come through have another understanding, and it seems to be this shifting baseline phenomena that's happening, which is that a scientist will start, and our longest standing scientist in this particular estuary has been here for 20 years, and he'll go, it was like this, it always was like this, we need to restore it back to that. But of course his memory is 20 years old. Whereas the local fisherman has been there for 4 generations, and he's been there all of his life, and he's 70, and his memory is different. And so there's a lot of arguments over what the estuary should or shouldn't look like. And so what I've been trying to do is piecing together what the original system looked like, or at least what it's looked like at different times over the last 100, 200 years. And I started by trying to gather together data on salinity. So here you can see some data on the salinity in the 1960s and some data on salinity more recently in this last decade. And you can see that it is, even though it's 10 kilometres further up the estuary, so it should be a lot fresher in the 1960s stuff, that it isn't. And so it's really quite interesting to look at those, but I figured that the biochemical and water quality data wasn't going to give me what I required to answer their questions. It just isn't the data. We're about 200 kilometres from the nearest city. What's that, 100 and something around miles? Yes. So I've started looking at the biota, so the plants and the animals and the algae that exist in the area. And what we've found, once again with it, is even though there is a lot of records, there's 70,000 records for the Coorong of what previously existed there, they've really only been kept since 2000 in digital record. what happened before is, you know, in the 90s we had 4,000, 5,000 records. In the 80s we had the same. But as soon as we get back to the contested time when we're not quite sure what the system looked like in the 50s, 60s, 70s, 1920s and so forth, there is practically no records. We have 3 or 4 or 100 or 200 records in each of those years. What's more, we tend to people, when they come into an estuary, tend to not look at the sediment. They tend to not look at the bacteria and the fungi and the reptiles and the insects and the algae that's in the water. People look at birds. And so we have this huge, as you can see in the top diagram, this huge amount of bird records and very little else, which really shows you what people were coming here to look at. And also, of course, the records all occur during the nicest months of the year. So it's nice to come out here over summer and spring and autumn, but in late autumn and winter, which it is here at the moment, and it's -5 degrees, nobody's out here looking at things. So this obviously makes it really hard to put the system back together. And this image hasn't come through. So we will just skip over that one. What I've been trying to do is fill up those earlier parts of the data set and those seasons that we don't have. So what I've been doing is going back as far as I can in the record and collecting data. Now, the earliest records that we have are the 1846 images that came from an explorer adventure into this area. There's also traditional owner stories as well. So looking at these and working out what species were where and what locations were. But there's also some really interesting things in the 1800s about the lakes. This is about a particular species of bacteria, cyanobacteria to be precise, that only blooms in brackish water. And it only blooms at brackish water right at the top of where animals like to drink it. And so the very presence of the bloom at this location tells us what salinity the water was. And yet the latest research says this lake was always fresh, and we know that it was at least seawater salinity. And so that's always fascinating to look at. I've been looking at these to identify plants and depth of water from the boats that came through, because we have the names of the boats that went through. Looking at the plants alongside this causeway. It's a very well-known causeway, trying to identify what they were in the early 1900s. And tying them to other structural changes. Tying the vegetation here to when the drainage went through and when changes in irrigation occurred. Phenomena like this that where the sand dunes have moved and you can see the mud putting up in front. We've been able to sit down and look at the photograph in detail, the original, and work out the type of mud that was there compared to what is there now. It's all black ooze now, and that it was obviously estuarine marl. Looking at known phenomena of breeding of birds, it's absolutely incredible. Looking at aerial photographs to see the movement of sand. This was the mouth in the 1940s, and ever since the barrages have been put in between the lakes and the Coorong, that the flow that comes down, the water flows through here, and 120 kilometres further south. As you can see, it's far larger in the 1940s than it is now. And our biggest find to date, and what's killing me at the moment as I sit here and type it up, is one fisherman came to me and he says, well, I've kept daily records, he says, for the last 20 years. Doesn't go back as far as you want. It turns out his father's got more records than his grandfather before. But this particular fisherman, just this one man, has kept records every day for 20 years. It's 42,000 records. Within which he's recorded 58 million biota, so 58 million birds, fish, algae, things like that, and over 1,000 margin notes. So this one is quite entertaining. He talks about catching a mulloway, which had a pair of sunglasses stuck over it, which he removed. But often it's records of really, really rare animals that have come through. And so that's my job at the moment, is just that data set alone will more than double what we know about the estuary over the last 20, 30 years. And I wish to say a quick thank you. This work has been funded by the Australian government in part through research funding. It's also been sponsored by the fishermen themselves, who are really desperate to get their fishery back, and supported by the Lakes and Coorong Environmental Trust. Thank you.